We're going to try and just see if this progresses well, which it does. Welcome to the ARC Training Centre in Predictive Breeding seminar series again for another amazing talk. We always like to start these seminar series by acknowledging country, and the ARC Training Centre in Predictive Breeding acknowledges the traditional owners and their custodianship on the lands on which we're meeting today. And we pay our respects to their ancestors and descendants who continue cultural and spiritual connections to country. And we want to just recognise their valuable contributions to Australian and global society. And I'd just like to extend this to say that we'd like to recognise all of the traditional owners of the land on where we all do our research, and where the people did the research on which we are building this knowledge. So not just recognising our own research, but those that came before us as well. So many of you may know me, but my name is Elizabeth Ross, and I am a Senior Research Fellow in QAFI, the Queensland Alliance for Agriculture and Food Innovation in the Centre of Animal Science. And I have been at QAFI for 8 years now. So it's been a really great journey to watch researchers come and go. And one of the great pleasures is the collaborations that we get to build working in this research intensive institution. And that brings me to where I am today, getting to host this seminar series with one of my collaborators. So just before we start and before I jump into the intro, I would just like to do a little bit of housekeeping. Today's seminar will be running from 1 to 2 o'clock. There will be questions, and we're going to hold those questions off until the end of the presentation. We are doing both an online and in-person event. So if you're online and you have questions, please just pop them into the Q&A section in the Zoom link, not the chat section. That will help facilitate making sure that all of those questions are addressed. And of course, there will be time to have a chat in person with James at the end. On that as well, just to let you know that the session is recorded. So if there's anything that is confidential, maybe better for a chat afterwards rather than on the recording. And that basically wraps up our housekeeping, which gives me the opportunity to introduce James Kijas. So James is a research group leader in CSIRO. He's an applied breeder and genome scientist working with the aquaculture industry. And he operates a range of commercial breeding programs in Australia and internationally. And recently I had the privilege to go to Korea, and the number of people that actually asked me, "Oh, you're in Brisbane. Do you know James Kijas?" "Oh, James, yeah, no, I remember James when I first saw him. He rocked up in the middle of Norway and he was wearing thongs and we all thought he was a bit nuts." But that is the kind of person that is really fun to work with. He doesn't take himself too seriously, but he does some really amazing science with a really sort of well-experienced and level approach to what he's doing. So with that, I would like to welcome James to the stage, and I really look forward to hearing about all of your work. So that is the clicking you want.
Great. Thank you, Liz. So thank you for that kind introduction, and thanks everybody for taking the time to come today and listen to, I guess, a domain of breeding that maybe many of you haven't heard a lot about, perhaps in the form of aquaculture. And thanks to everybody online that's joining today to listen as well. So yeah, as it says, aquaculture, breeding, genetics and genomics. So to kick things off, I thought, as Liz mentioned, I work for CSIRO. I've worked for CSIRO longer than I care to admit. But I thought I'd start with just sharing a little about the group and what we do. So as Liz mentioned, we operate commercial applied breeding programs across aquaculture nationally and internationally, and we consider ourselves as applied scientists. So we're really embedded with clients, aquaculture enterprises, to understand their needs and where applied breeding and genomics can make a difference to what they do. We work with them lockstep to deliver impact. So the sorts of things that means in practice is the commercial delivery of breeding values, or EBVs, and in some instances, genomic breeding values, GBVs. We look to implement genomics improvements to speed genetic gains in those breeding programs. We're quantitative geneticists, and we spend a lot of time with data systems, as I'll talk about at the end of the program, of the talk this afternoon. So the group that I lead has staff in 3 places: here in Brisbane, in this building, an hour and a bit up the road in Bribie Island, for those of you that know where Bribie is, where we have a research facility. So we've got animals under management there, primarily for production science and nutrition. We do a bit of genetics there, but not much. And then down in Hobart, we have a big team of quantitative geneticists that face into the aquaculture industries in the southern part of the country. So in the table, I've listed out the species that we have applied breeding programs for, where those programs are located, and the duration that those programs have been in operation with CSIRO. So slides a couple of years out of date, so you can add a couple of years to those. So you can see there's a real diversity of species there. There's a bunch of oyster programs. The bulk of what we do is finfish, so things like Atlantic salmon, tri-catfish, which if you go into a supermarket in Australia is marketed as bassa, barramundi and Murray cod. The other species that we work with a lot is shrimp or prawn. I'll use prawn and shrimp interchangeably today. 2 species of shrimp, white-legged shrimp, its Latin name is Vaname, and black tiger shrimp or prawns, which is the one cultured in Australia, and that's Penaeus modernon. So we do a lot with shrimp. So there's some pics of the team. And I put this up to make the point that applied breeding and the delivery of commercial breeding programs needs a bunch of capabilities and expertise beyond the ones that you might imagine. So, at the top there, we know something about animal husbandry, genetics, and biology, but there's a bunch of skills that we bring to get these activities off the ground. I'll highlight a couple: people skills and consultation. So we actually listen to our clients and what their problems are and how their organizations actually run and what the efficiencies might be, rather than just assuming that we know what we're talking about, because often we don't. And people skills, because these are commercial entities, they've got their own competing priorities, and what we might like to do as applied scientists and animal breeders isn't always the top priority, in these commercial entities, so that there's a bit of give and take. One capability that isn't actually on there that we're increasingly doing is developing traits with, for example, image-based capture to measure what were hard to measure traits and turning them into easy to measure traits, phenomics activities. Okay, so the outline of today's talk is I'll give you a really high-level view of the anatomy of aquaculture at a global level. The opportunity and the challenge, they're often the same thing. And then I'll bring it down to, well, what does aquaculture look like in Australia? Before I go into the main body of the talk, which is to try and convince you that even entry-level genomics can have a big upside for aquaculture genetics. I'll step you through a couple of genome-wide association studies, or GWAS studies, that we've done, and explain why that's assisted us with our applied breeding activities. As I'm sure you're all familiar with genomic selection, so I'll give you a case study in genomic selection in aquaculture. And then, I forgot to put on the timer. If we've got time at the end, I'll make the case for data systems and how a data system ties everything together when we're delivering these programs. Okay, so what does aquaculture look like in progress? So I'll give you 2 examples, quite different examples, for what aquaculture can look like in Australia. So on the far side of the slide there, my colleague Tanzan is releasing some juvenile tiger prawns into that big pond. You can see in the bottom there she is standing in the pond. And so we operate extensive tiger prawn activities in Northern Australia. So I put on the slide that they're low input. So they're low input in terms of the stocking density of prawns that are going in there. So we stock them at really low densities, like 2 animals per square metre, which is a 50th of what a semi-intensive or an intensive farm would do. And the reason is that we don't need other inputs into that farming system. So we can operate and grow our animals in these ponds without electricity for aeration. We don't even have to feed the animals, because the natural productivity of all the little critters that grow naturally in the pond become the food source for the animals. We don't need skilled workforce. So this is a place-appropriate farming method for Northern Australia. You can compare that then with the side of the slide closest to me, which is the Tasmanian Atlantic salmon industry. So we go from the top of the country to the bottom of the country, and it's completely the opposite. It's very intensified, high input in these large indoor tanks. You've got really high biomass, huge numbers of fish that they grow out to a size before they put them into their sea cages. Takes a lot of energy, but high control systems, completely different to the other aquaculture system that I've shown. So there's also, not only is there diversity in place and species, there's also quite a diversity of production systems in aquaculture. So you can see my colleague Dan Richards, who's the owner of Humpty Doo Barramundi, the biggest barra farm in Australia, and he's standing by one of their earthen ponds, so that on-land ponds that they operate in Humpty Doo, just outside of Darwin. So that's a reasonably low-cost production system where you can see a paddle wheel in the foreground there. So it requires electricity to oxygenate the water because of the biomass under management in the pond, and their production costs are really staff and fish feed. So comparatively low cost. But there's also not so much control in an open system like that. So these are outdoors. It's comparatively harder to control pathogens and water treatment, and yeah, they're natural outdoor systems. Compare that to this one up the top. So that's a recirculating aquaculture system, or RAS system. So these are completely different in so much as they can operate on zero exchange water. So the water's in these tanks. It gets pumped out of the tank, it gets filtered and cleaned and returned to the tank. And so the water just goes around and around. You're not drawing in water, using it once, and then it's leaving. You're recirculating it. And so these are becoming very popular because you've got absolute control over the production system. High biosecure operation because they're indoors. You're not pumping in water from the ocean or a creek or something. You can control the water temperature. You can control the water quality. So they're great systems. The downside is they're very power hungry currently. So the cost of production is really high. So they work pretty well, but they're difficult to scale and keep economically viable. So what many industries will do, the barramundi industry included, is you grow animals to a certain size in RAS systems, and then when they're, say, 100, 150 grams, you can stock them out into your either sea cages or in-ground ponds. So hopefully that's just giving you a feel for the sorts of systems that are in use across aquaculture. So we'll change gears now and take a global view of aquaculture and how it's different from, for example, the livestock industries. So a couple of points there. The first is there's this enormous diversity globally of species that are under management and being cultured in aquaculture. So if we think about, if you look at the top here, this is the global meat production as a percentage split across species. And there are 3 species that we rely on for about 80% of global meat production. They're not going to surprise you: chickens, cattle and beef. Sorry, pigs, chickens and cattle. 80% of global production. So to get to 80% of global aquaculture production, you need to bundle again together not 3 species, but 70 species. So it's incredibly diverse. Where is aquaculture occurring? You can see in the bottom here, like production in Asia absolutely dwarfs the rest of the world. Like, it is just a massive industry. It's primarily finfish culture, which you can see in the blue bar there. And there's a bunch of crustaceans, which is primarily shrimp aquaculture, and crabs, but dominated by shrimp. And so aquaculture productivity in the rest of these major regions of the globe, as you can see, is much, much lower. And maybe I think of the most relevant to us as geneticists is that these aquaculture species are really recent domesticates. So again, comparing with, say, sheep and cattle, we're talking about 10,000 years ago when they were brought into captive management and some level of domestication occurred. For these major production species in aquaculture, we're talking 30 years. So the Atlantic salmon industry, which is, a major economic driver in Northern Europe, in Norway in particular. Domestication started in the late 1960s, early 1970s. So for an animal with a 3-year generation time, that's not that many generations since these things were, swimming around as wild animals. So that there's an amazing potential for us as geneticists, because we've really only started out with these species to bring them under captive management and apply advanced breeding. So there's a lot of upside in terms of breeding these animals for productivity. So one more then around sort of these global trends in aquaculture. So the slide on the far side, I'd say far side or near side because left and right has always eluded me. But anyway, so this one over here, so this shows productivity in terms of global production in million tonnes across time. And you can see that the proportion or the amount of productivity that we get from wild catch, so going out into the ocean, has plateaued. Maybe no surprise, the oceans of the world can only supply so much, seafood. So beginning in kind of the late 80s, it's plateaued, and that's when aquaculture has just absolutely exploded, and it continues just to ramp right up. So where is that expansion coming from? You can see in the different colours there. So this blue is freshwater aquaculture. So that's really been the major driver of this huge expansion in aquaculture production. So we're talking species here like tilapia, the tra catfish or bassa I mentioned. There's a number of species of carp. And these have been so successful because they're low trophic, they're omnivores and herbivores, so that they've got a really low cost of production, and so you can ramp it up and scale it and make it profitable. So this down the bottom makes the same kind of point. So these freshwater aquaculture species, lower kind of price per kilo generally, compared to marine aquaculture, which is salmonids, Atlantic salmon, pink salmons and things. They get you more money per kilo, but the cost of production is higher. All right, so one slide on where Australian aquaculture is. So in 2021, this is data from ABARES. It's a $3 billion industry, and it had plateaued. Plateaued primarily because the salmon industry had reached kind of a maximum level. And so if you split the production between aquaculture and wild catch, a couple of years ago, aquaculture wasn't at 50-50. We've now passed that 50-50 point, and you can see the estimate is in a couple of years' time, it'll be 2/3 production from aquaculture and 1/3 from wild catch in Australia. And I was surprised to see that our per person consumption of seafood is at about the same rate as cattle, and that surprised me. Like when I was a kid, it was a while ago, but I reckon cattle would have been twice the consumption of seafood. All right, so that gives you a snapshot of where things are at. And now I'll step through a couple of high-level challenges and opportunities. And this is a challenge that might not seem obvious from the get-go, but just domesticating aquaculture species has proven and continues to be a major challenge. So there's a paper here from 2021 that defined 5 levels of domestication. So if we go all the way to the bottom of that table at level 1, you might think first trials where we're acclimating a species to captive management. Let's bung some animals in a pond or a tank and let's see how they go. So at that point, when that review was done, 39 species were at that very bottom level. If we go one up to level 2, part of the life cycle of the species is in captivity, but part of it is still in the wild due to bottlenecks, reproductive biology and disease bottlenecks generally. So that is the case for currently in 2025, the Australian tiger prawn industry, which are these photos at the bottom. So every year, and it's happening right now at this time of year, prawn growers charter these trawlers. They go out into the northern prawn fishery out of Darwin, and they catch a couple of hundred mature broodstock animals. They bring them back on land into hatcheries, they condition them, they spawn them, and then the juveniles are what they stock out into their grow-out ponds to produce harvest-sized animals. So that's a great way to introduce diseases into your farm every year, because you're just going out and what do you get from the wild? You don't know. And it's a sure way to lock yourself out of any genetic gain, because you're going back to generation zero every year. We could talk about, well, why are they still doing this? They make money doing this, and domesticating this particular species is hard, but there's a massive opportunity there. So if we go up to the top of the slide then, at level 5, we've got selective breeding programs underway and a defined breeding goal that's generating the juveniles for grow out. And these authors reckon there were about 30 species at that level. So not that many given how many aquaculture species there are under management. So that's a challenge. It's also an opportunity. So I spent a bit of time on this one. So this comes from a review in 2020 by colleague Ross Houston and his team at the Roslin. So they had a look at the top 10 producing aquaculture species globally. So we'll start with our friend the grass carp here, nearly 6 million tonnes annually, down to Atlantic salmon, which is around half that. And then putting against that, well, what is the use of genetic technologies in these industries? So got that at 4 levels, where the lowest one now is level 1. Juveniles are supplied from wild sources, hatchery applied. They don't do any genetic management. That's basically what I just described for the Australian tiger prawn industry. Or at level 2, there's some basic genetics screening and selection in the hatchery. There might be some genetic diversity or inbreeding management. So that's level 1 or 2. And that is the 2 of the 3 biggest production species on the planet. So there's a massive opportunity in these carp species. So if we go down the levels then, either juveniles are supplied exclusively from breeding programs at level 3, or you've now implemented more advanced breeding programs at level 4. And level 4, for these top 10, there's only Vaname, shrimp, tilapia, and Atlantic salmon. Okay, so in practice, how do we go about implementing genetics and genomics to improve aquaculture species? So in this table, I've listed down the side a bunch of activities. What do we do in practice? So it could be things like parentage assignment, inbreeding management, doing genetic diversity audits of founder broodstock to figure out if we've got enough genetic diversity to support long-term genetic gain, GWAS to link genotype and phenotype, pathogen detection, and at the bottom there, genomic prediction. So for each of those, I've listed the application domain. Where would you deploy this activity, and what are the genomic tools that you would need to support those tools, reference genomes, SNP tools and stuff. So I'll use this as a structure for the rest of the talk to, first of all, talk about barramundi and try and convince you that even entry-level genomics can have a big impact on what you want to do in breeding. Next, I'll talk about Atlantic salmon and shrimp with some GWAS studies we've done and why you would want to do that to inform the way forward for applied breeding. And then down the bottom, genomic prediction. I'll skip through a case study in genomic prediction for Atlantic salmon. So let's talk then about entry-level genomics and barramundi, which is a species close to my heart. A colleague said to me, this barramundi actually looks like a cardboard cutout. I can assure you it's not. It's just taken with a pretty low density camera, phone camera in the field. Anyway, so before we start talking about barramundi and breeding, there's a couple of features of the biology of this creature that it's important to mention. So it's endemic throughout Asia. It's called Asian sea bass in many places, barramundi in Australia. So it's a social spawner. So in the wild, males and females aggregate and they spawn socially. And here's one that will do your head in if you didn't already know. They undergo a male to female transition. So they come into sexual reproduction first as males, and then nearly all of them, not all of them, but let's, for the purpose of this discussion, say all of them, when they're older and larger, they transition into females. So if you're off a cattle farm and used to, breeding animals, use rams, well, it does complicate life when your ewes turn into rams, and you're an animal breeder, I can confirm that. They're cannibalistic, big ones eat little ones. They're highly fecund, so you can generate hundreds of thousands of progeny from a single cross. And they're extremely robust. They taste great, but from a production perspective, they also will thrive in anything from fresh to salt water, a range of abiotic conditions in terms of dissolved oxygen, water quality. They're really hardy fish, and so as a result, they continue to grow in popularity as a farmed aquaculture industry. So these quirks of their biology have real life consequences if you want to set up and operate a breeding program. So because they're a social spawner, you've got reduced reproductive control. We can't use AI in the species because we can't externally fertilize them. So we can't do one-to-one matings as we would like. So that's a problem. The male to female transition, as I said, complicates broodstock management, but we can manage it. Because they're social spawners and because they're cannibalistic, we can't hold animals in family groups after spawning until they're big enough to stick a tag either on or in them. So we're forced to do communal rearing across families in their early life. We've got no choice. And as I mentioned, because they're highly fecund, you have to manage inbreeding. So you could run a commercial grow out with not many broodstock. So you have to keep a handle on inbreeding. And so to do that, a SNP tool is going to solve a whole bunch of these problems, or these challenges, I should say. So in the next couple of slides, I'm going to have one slide on how we went about generating a first generation SNP tool to get cracking with applied breeding in the species. And then I'll step you through where we landed for a design and the operation of a breeding program in the context of that particular biology, because that's kind of what the team does. We design and implement breeding programs. So we'll step through one today. So to get us going, we wanted a really low resolution SNP tool to get us started. And that was really around cost. We needed something that was cheap to develop, like it had to be really cheap. And that was because of the, I won't get rid of it. Had to be informative in the target population. I guess that's pretty obvious. And it had to be capable of supporting early breeding applications. So we ended up sequencing only 10 commercial broodstock, founding broodstock from the breeding program. We used some database genomes that were publicly available. We went and identified 4 million SNP, but we only formatted 340 of them on a technology for routine genotyping. And this was to control cost. So we used the CASP, or competitive allele specific PCR technology, which was made available through our partners at the Center for Aquaculture Technologies, or CAT, in Southern California. So to get cracking, this is a couple of years ago now, we simply asked the question, well, will 340 SNP deliver you parentage? So we took a set of 94 progeny from a group spawn event, and we set 125 candidate, or we declared 125 broodstock as possible candidates to be declared the true parents using the SNP genotypes. And indeed, it worked really well. It delivers parentage with high accuracy. So even a modest number of SNP, if they're well designed, will tick the box for parentage. So that tool has now been in routine operation for a number of years, and it does what we need it to do in a breeding context. So what does a breeding program look like in practice? So I'll spend a bit of time on this because there's a fair bit of detail. So what you're seeing there in the schematic is a single cycle of the breeding program. So if you look at the top here, so at sort of 12 o'clock, if this was a clock, we do a group spawn event. So typically we might be spawning 15 to 20 animals, male and female, in an event, and we'll generate a variable number of families, but typically it'll be around 30 families to start off a breeding cycle. So as I mentioned, those families are raised communally in, this is the stage 1 nursery at Humpty Doo Barramundi. So the animals are raised in these tanks until they're large enough that they're stocked out for growth in these outdoor earthen ponds. And from a single group spawn event, we stock out 3 commercial ponds. So that's what these 3 lines are. So there's 3 cohorts per cycle. and so cohorts are nested within cycle. So one of these ponds is harvested at 9 months of age, and they become a plate fish. So a 9-month-old animal is typically 900 grams to about 1 kilo. And all of the animals from that pond are harvested. And at that point, we progeny test animals from all 3 cohorts, because this is the last time in a breeding cycle that we can measure progeny test from all of the cohorts, because one has just been harvested and gone off to market. So at this point at night, so we collect, I should say that the objective for the breeding program is currently only growth. So we're considering turning it into a multi-trait program, but for now, the sole objective is growth. So we measure our animals at 9 months of age, wet weight, but we also do a second thing. So it's at this point that we select animals from the fastest growing pond. We take them out of that production pond. We put a microchip in them, like you might put in a pet dog or a cat, so we know who's who. We take a DNA sample from them. That's what's happening down here. And then we put them into a separate management unit on the farm so they don't get inadvertently harvested and sold and somebody eats a pit tag, which wouldn't be good. So then we've got our broodstock candidates held in a separate place on farm. The cohorts go through to 15 months of age when they're all harvested for commercial sale, and that's a fillet-sized fish, 2.5 to 3 kilos. So if you buy a Humpty Doo barramundi fillet in a supermarket, it's probably approximately 15-month-old fish. Now, in the time between the 9 months and 15 months, we've taken our DNA sample, we've done our genotyping, we've established the pedigree of the broodstock candidates, we've generated our estimated breeding value for those candidates, and so we can do a broodstock sort here at 15 months. I forgot to mention that we tag and hold many more broodstock candidates than we know we're going to use. So we're able to apply selection pressure here at 15 months, and we'll keep just a fraction of the animals that we tagged at 9 months. Some will go into storage, if you like, and contribute to a future cycle. And so these are animals we're warehousing and holding on to serve as females in a couple of years' time. But a bunch of the animals that we've tagged at 9 months will also go forwards straight away into a conditioning and a mate allocation process to complete the cycle, and they'll spawn again to start the next cycle of the breeding program. So we don't just do this once a year. We do this 6 times a year. So there's just this rolling cycle of progeny testing, broodstock culling, and group spawning. All right. So what I'll do now, so that completes I've hoped I've convinced. I'm not going to show you any genetic gains plots and things because that's commercial in confidence, but I can tell you it's been operating since 2018 and it's working very well. All right, so next up, a couple of examples of genome-wide association studies. So why would we do that? It's really useful to know the genetic architecture of any given trait that we're trying to improve through applied breeding. So is the trait A, controlled by a small number of genes of really large effect, or B, is it highly multigenic and lots of genes of individually small effects? Because that will calibrate how you might approach making genetic progress. So it turns out in aquaculture species, there are still big genes of effect that are out there. So salinity tolerance in tilapia, there's a gene explaining 80% of the phenotypic variation. I mean, that's just ridiculous. Famous example of infectious pancreatic necrosis or IPN resistance in Atlantic salmon. Again, a big whacking amount of the variance explained by a single gene. And I suspect, remember I said towards the beginning, these animals are recent domesticates. So we just haven't had time, I suspect, to have bred these beneficial alleles to fixation like we might have in other species. Okay, so that guides what you're going to do. So for these two, marker-assisted selection, which has got a pretty bad reputation, I have to say, it actually does work where, traits have got this sort of architecture. But as we know, the majority of the traits that we'd like to improve are highly multigenic, and we'll get onto genomic selection in a minute. Okay, so two GWAS, both related to sex, and sex really matters in aquaculture for a couple of reasons. In Atlantic salmon, it matters because males in these grow-out production systems mature quicker than females. So the box plot there shows at 23 months of age in the Tasmanian freshwater systems that we operate, males are a full half kilo lighter than females. That's not good. The other thing is because they mature earlier, when the males mature, their energy budget changes and they allocate energy into gonad production and their fillet quality disappears, and so they're almost unsaleable. It's an enormous production challenge, which will be helped or is helped by understanding the genetic basis of sex determination. Switching over to shrimp or white-leg shrimp, Vannamei, it's the other way around. Again, females grow faster, and this is true in a bunch of Penaeid species, but it's reversed in some other species. You can see the graph there. Females grow faster. So again, we would like to be able to control sex determination. To do that, we need to know how it works. So as mammals, things are reasonably straightforward. SRY gene sits on the Y chromosome. Y chromosome confers maleness. That is not the case and this is just finfish. So that table lists all the master regulators of sex determination across species and other, well, not just fish, and other organisms. And pretty much every player in the estrogen and androgen control cascade that regulates ovarian and testicular differentiation is a master regulator of sex in a species if you look hard enough. So if your species of interest in aquaculture doesn't have the sex determination gene known, you can't guess what it's going to be. It could be anything. So going back to our friend salmon, so by the time we had access to a 45K SNP chip, we knew maleness associated with the presence of the SDY gene, and we'd collected nearly 500 families of over 3-year classes of fish, and we had phenotypic assignment of sex based on external phenotype, coloration, and the front of their jaw, basically. And so we performed a GWAS to see where in the genome the genetic determinant ends up, naively thinking maybe it was going to be in one place. Nah, it's all over the place. So at a population level, you see association, really strong association to 3 chromosomes and some other places as well. In the interest of time, I won't go into explaining all of it, except to say that if you look within a family lineage, the genetic determinant resides on only 1 chromosome. But at a population level, the SY gene can be in different places, which explains why our DNA diagnostics weren't working very well. And even now, our DNA diagnostics are not 100% predictive. And this is a real problem for the industry, because what they do to create all female commercial animals for grow out is they start with what they hope is a female, and you can hormonally treat it to turn it into a productive male. And if you cross one of these hormonally induced males with a normal female, you should get all female offspring. Except if the animal that you hormonally induced actually wasn't female and it was male to begin with, you can do all of the tricks you want and it's still going to produce males. So if you don't have a rock solid DNA predictive test, you're still going to run into males appearing in commercial populations. And that's unfortunately where we still are. And there's a suspicion that sex determination in this species isn't under 100% genetic control, that there is a minor contribution of environment. So I'll leave salmon there, talk briefly about tiger prawns. So Penaeus monodon is the tiger prawn, is the species that we grow here. We were interested to know what the sex determination gene was. There are now 2 reference genomes resolved to chromosome level. The second of those that you can see, Hulaman et al. 2021, sequenced over here at AGRF. And there's a paper from 10 years ago showing that there was a single associated region in the genome to a linkage group. So it didn't have a chromosome assignment because it predated the reference genomes. So to make progress here, we developed an 8K, 8,000 strong SNP array, Illumina array to support a bunch of breeding applications for clients here and internationally, and that paper's under review at the moment. And we genotyped 113 males and 170 females And you can see it's a much clearer picture here. We see one strong association signal. We see some outlier SNP, which when I track them down, that they're actually assembly errors rather than true association signals elsewhere in the genome. So we see very strong linkage disequilibrium across the 4 megabase region of the prawn genome. Perfect association between segregation at SNP and the phenotype. So that's great for what we want to do in our breeding programs. Looking into that 4 megabase region, we don't see any obvious genes that would confer sex determination. And that's an active area of research that we're chasing that down to a causal mutation as a target for future gene editing, which we're also working on. All right, so last up then, genomic prediction. So again, I'll stick with Atlantic salmon that we've done a lot of work here. So I know you all know this. There's someone in the room that would know this better than anybody. So I'll step through it very quickly, and then I'll tell you why this is particularly a compelling paradigm for aquaculture. So at the top there, we've got our animals that we've generated from mating, fish in different colours for different families, and we put them into 2, we'll call them tanks. We manage them as 2 populations. So the one here, the selection candidates, we put a pit tag in them and we genotype them. The other ones are our reference population. We tag them and we genotype them, but we do an additional thing. We phenotype them. We phenotype them for all of the things that we're interested in. And this has one really important advantage that I'll get onto. So what we can do in our reference population is we can, for example, expose them to live pathogen and assess, their ability, a metric like survival. So we can do that here, and the animals that we're going to use as our breeding candidates, we can keep as a biosecure population. So after we've done our genotyping, we can create our genome relationship matrix, which gives us really fine-grained understanding of the relative relations of pairs of individuals in both of the populations, candidate and reference. Build our genomic prediction equation. So for each SNP, what's its contribution to trait variation? Create our GEBVs, and then we can select those animals that we want to take forward to mate for the next generation. So as I've said, the real advantage for this in aquaculture is disease challenges while maintaining biosecure operation, particularly in the prawn shrimp world. These animals are highly sensitive to kind of a dozen pathogens that will just knock them over in a production setting. And so you have to maintain the breeding animals disease-free. But using this paradigm, we can still collect disease data in the reference population. The big advantage for us in aquaculture is because of their fecundity, we can generate hundreds of thousands of animals for progeny testing really quickly and cheaply. So we don't have to just make one reference population. We can make a bunch of reference populations using the same genetics going in at the top. So we might have a reference population to explore performance in low dissolved oxygen environments, low salinity environments, high temperature, low temperature. So we can do all of those things and create these reference populations reasonably cheaply and quickly. And you'll be familiar with the other reasons that drive genetic selection. So does this work in aquaculture? You bet it works in aquaculture. So this review from 2020 did a meta-analysis of a whole bunch of studies. So you can see 14 studies for growth and 12 across 12 species, 23 studies that looked at disease traits from 11 species, and on average, something like a 25% relative increase in accuracy moving from traditional breeding to genomic selection. So that is just a massive increase. A note of perhaps caution that the reference population sizes, densities, and there are all sorts of variabilities between these studies, but the take home is, yeah, absolutely this works in aquaculture. So why isn't everybody using it? Seems like a no-brainer. Well, there are some challenges. You need a SNP tool, and as I've been banging on about today, there's a lot of species here that don't all have SNP tool resources. So our group has generated a bunch of SNP platforms for different species. Managing cost, the entry cost and the ongoing cost of genotyping means the value proposition may or may not be there. So there's a couple of tactics that we can use to manage the genotyping cost, which we can talk about afterwards if you're interested. You need a data system and skilled practitioners. The volume of data that you generate quickly can overwhelm you, and so you need some skilled practitioners to assist you with that. And this is really challenging to implement for enterprises that don't already have some strong culture of using data to make decisions. So if an enterprise is already generating breeding values and they're using them for their selection, it's comparatively easy to advance them to genomic prediction. But if they're not already, measuring a bunch of stuff and using it for management, it despite often high levels of enthusiasm, it can be a challenge. So then 2 slides on a case study. This is the Atlantic salmon industry that we've been running with our colleagues at Saltas for many years. Switched over to genomic prediction in 2018, and in 2021, my colleagues published this paper about the gain realised through the implementation of genomic prediction. So you can see in the table there's a bunch of primary traits, secondary traits and monitoring traits. You can see the relative selection pressure in the index there. So we're applying pretty high pressure for a disease resistance trait to amoebic gill disease and harvest weight, and then less so on these other traits and just a monitor trait to make sure we don't go back on fat. So sorted ourselves out a 50K tool, which was informative in our particular strain of salmon in Tasmania, built the reference population, modified our data system, built a routine GBV pipeline. And so what has that done? So this is the wash-up. So genetic gain as expressed as the percent per year for these traits. You can see the numbers where we got to in 2018 after about 15 years. That was the average per year, and then the jump after implementation. So, that's a significant improvement. And just at the moment, we've got an external consultant in to do a cost-benefit analysis of how successful that's been in financial terms. And we haven't completed that, but yeah, it was a compelling value proposition to do it. All right, so I think I've got a couple of minutes left, and I live and breathe this stuff, and so I'm going to try and convince you that data systems are important and good. It's not everybody's cup of tea. I'll admit that from the get-go. So when I say data system, what is a data system? So I think of it's got 3 components. It's got a database to house the streams of data that we're collecting. It's got hardware for automated data capture. So the days of us standing around with waterproof paper and pencils and yelling out like weights, nah, not anymore. And it's got to have decision support features for our clients so they can automate what they're doing and how they're doing it in a breeding context. So the middle one there, hardware for data collection. I'll just give you a feel for that. So this is a broodstock tagging event at 9 months at Humpty Doo. So what we're doing is we're putting in a pit tag, that micro chip, reading it off with a pit tag reader here, and that's going straight into the data system. So this box here, it's a Pelican case with a computer and some dedicated software in it. So when a fish is anaesthetised, it gets a pit tag put in it. We're taking a tail biopsy into this closed tube, which you might be able to see there has got a 2D barcode on the end of it. So we've got a handheld scanner that gets collected. We're getting a fin clip in ethanol, sits on a shelf for when we can whole genome sequence animals for 10 cents. Or have we got any samples from those animals? Yes, we do, because we've collected one. We're automatically collecting weight and length. And it all goes into this bit of software. And when we're happy we've got all of the data from that fish, we commit that record, and it creates a data string where it knits all of those bits together so that we don't uncouple a sample from a pit tag. And so we're always sure that our DNA profile from an animal matches the pit tag, because if that ever gets uncoupled, game over. So the database, another part of the system, of course, I think we've got something like 2 billion genotypes and 7 million records in it across the breeding programs that we manage. So you need a pretty good database and software engineers to run it. And then the last part is functionality. So we've got automated, systems when we get a dispatch of genotyping, it gets sucked into the database, we press a button and we do our quality control reports and filtering and things. So we're not doing this stuff manually anymore, but we're getting reports so we can check on the quality and how it's going. And we've also in the data system got a bunch of breeding tools. So one is optimal contribution, where we're managing the competing drivers of inbreeding management and genetic gain. And that in real life turns into, for example, lists that we can send to our clients to do keep culls on broodstock sorts and actually breed the animals. So this is the last slide, and this is what ties it all together. So I showed you, and we went through that single cycle of the barramundi breeding program and each of the events that occur there: broodstock tagging, broodstock sorting, spawning. So as that's going through the cycle, there's this dynamic process of information flow where we're collecting the data on farm, it's going into the database, we're doing analysis on the database, and we're sending information back out of the data system to the client so they can manage the animals in the field. And it's going on every day. All right, well, that is it. Two last very general points. Aquaculture is growing. It's playing an important role in food availability globally. And genetics and genomics, I hope I've convinced you today, is making an impact. And with that, I'll say thank you, and thank you for listening, and I'm keen for questions.
So we do have a microphone for anybody in the room. If you'd like to ask a question, just raise your hand and we'll take the microphone to you, and then we will alternate them with the online questions. So I'll read out any online questions, but we have one in the room first.
Good presentation. Thank you. From pig breeding, I know that there's quite a bit of G by E or G by M between pigs, the seed stock managed, at the seed stock level versus commercial production. Are your reference populations managed the same way as the commercial production? Or if not, do you know how much G by E or G by M you might see?
It's an excellent question. So we try and generate reference populations under what we call commercial-like environments for shrimp breeding. Sometimes you can see a divergence between the genetic gain and the phenotypic trend. And so you might wonder, well, why is that? Is that G by E? A genetic trend says that the animals should be performing at a particular level, but we might see a gap in performance. When we try and unpick that, it can be for a variety of reasons. The producer has been holding animals back by not feeding them as much because of market conditions. They might have overstocked, there might be disease. So there's a bunch of stuff that goes into it. But yeah, we certainly consider that when we're setting up our reference populations.
I might read out one of the online questions now. So the question's from Sebastian, and Sebastian says, "Hi, James."
Hi, Sebastian.
In our barramundi case study, you mentioned the breeding program had a strong focus on growth. This makes sense, bigger fish, bigger financial value. But can you please expand a little bit more on the relationship between growth and that male-female transition you mentioned? For example, do larger fish become female, or what drives the transition?
Wow, that's an excellent question, Sebastian. I wish I had a comprehensive answer for you. So we don't know the exact drivers of the transition. I sort of alluded to the fact that they nearly all transition from male to female, but they actually don't all transition. We certainly see an effect of farm-raised fish compared to wild-caught fish. Wild-caught fish will remain as males to older and larger sizes, whereas our farm-raised fish have a kind of precocious feminization where, because we're feeding them to promote growth, I suspect they do an energy budget and they go, I'm in good condition, I can commit to transitioning to female earlier than wild-caught animals. There's been some tremendous work done by colleagues at JCU looking at the epigenetic drivers of the change, but the actual triggers that set it off, we don't yet know.
Excellent. I think we have a question down the back by Ben.
Yep. Hi, James. Nice talk. So you describe your implementation of genomic selection where you got your reference population and then your breeding population. In the aquaculture species, is there anything you can do to crank the generation interval turn over the breeding cycles faster.
Yeah.
Like in plants, you have speed breeding, for example.
Yeah, well, the one example is in Barra, where there's a lot of work going into actually getting males and females being able to breed at about the same age. So bringing younger females. Other than that, I would say other things are on the to-do list prior to that speed breeding approach. But yeah, we are aware of it, and I think in time it'll pay off.
I'll read out one more online before we switch to another one in the room. So it's Sebastian again, says, "Hi, James. Could you please provide us any information on the ongoing eel aquaculture breeding efforts in Australia? My understanding at the moment is that all eels grown in aquaculture are not bred in hatcheries due to their complex life cycles."
Thanks for the question, Sebastian. I don't know much about eels. I didn't actually know we had an eel industry, I must admit. I know it's a big industry in Japan. But yeah, I'm afraid I can't enlighten you anymore on eel hatchery practice.
Maybe Sebastian can enlighten us at some point.
Absolutely.
Is there any other questions from anybody in the room?
I've got one, but if anybody else has one, they can go first.
You can ask your question. Everyone else can think while you ask it.
Okay,
How's that?
let's do that. So you mentioned very, very briefly gene editing, James. In aquaculture species, can you do that in the eggs?
Yeah.
Or do you have to produce stem lines?
No, micro injection direct into eggs is the way we're going. So that's in routine use for salmonids, for venomae, and we're having a go at it in monodon because it'll just be a game changer. We could spend all day talking about the regulatory environment and the social acceptance space, but the regulatory clarity is coming, I think, particularly in Australia. I'd say there's a very good chance in parts of the world it's already done and dusted, and edited animals are in the food chain in aquaculture species, absolutely. So yeah, we look on the technology cautiously positively. It's pretty challenging in prawns because the micro injection, the eggs are really hard, and so there's other quirks of the biology that mean it's difficult. Something like Atlantic salmon and salmonids, it's really good because you can externally acquire the gametes and they're reasonably easy to work with. So again, as I was saying, for breeding applications, the biologies of the species has got positives and negatives. It's also the same for PGE.
Any other questions in the room? I might quickly jump in with one if that's all right, James. You talked about the cost-benefit analysis that's been done and it looks really promising. And one of the things that we know about production industry is there's a lot of price sensitivity around how much this costs. Do you think with those sort of cost-benefit ratios coming out, there'll be a little bit more flexibility in people being willing to take on the price of genotyping?
I'd like to think so, but perhaps paradoxically, you do one of these cost-benefit analysis and it looks wonderful, and so it sets an expectation that this is absolutely what's going to happen. So it can be good or bad, but there's no doubt in industries of sufficient scale that are already ready and able to implement it, that there is a positive value proposition there. It really comes, so you can do in these cost-benefit analysis, as you may know, you set up the model and then you do a whole lot of what-if sensitivity testing. Well, what if our production was 10% more or 10% less? What if our cost of production changed? What does that do to the value proposition? And the one thing that really drives that positive value proposition is the scale of the industry, the tonnes produced of the product for sale. So enterprises will get to a point where applied breeding, just doing any breeding, has a positive value proposition. And it's the same for genomic prediction we see. So industries have to be at a significant scale. They have to accept that the cost of genotyping just becomes the transaction cost of running their business, but that there will be an upside. But it's not for everybody, but in some cases, I haven't seen a negative outcome from a cost-benefit analysis in an aquaculture industry. Maybe that's because if it wasn't going to be positive, we don't do the study in the first place.
Excellent. I think on that note, we might all thank James.
Thanks for coming, guys.
And just a few little mini housekeeping things at the end. The seminar series are all up on YouTube. So if you have missed any of them, please do log on and have a look at those. For those in the room, please join us and especially James for some snacks and some coffee afterwards. And keep your eyes out for the next announcement for the next seminar series, because I think it's really nice to be able to have these in-person events and get together and discuss some science. So thank you everybody for coming, and thank you, James, for giving us your time.